what makes this award very special? I know many of you here, and I've known you for a long time. I've met you at this meeting. I've met new people at this meeting this year. And that this award comes from you, people whose science I know and I admire so much, makes the award so very valuable. Science is a very social activity. And the things that we do, our successful experiments that we may do and spend hours in the lab, really assume meaning because they're integrated into a broader community that we all participate in. And so in thinking about what I would talk about today, I decided I wanted to talk a little bit about ideas in the scientific community, but actually specifically on maternal and zygotic activity in early embryonic development. But I wanted to put this in a personal context by describing to you two of my personal scientific heroes, and in some way talk about their experiments, but how they relate to my own science. My first hero that I'd like to talk about, and I actually have many more heroes, but these two are really good. First is Theodor Boveri, and I'm going to extend, you can see, try to extend this Society of Developmental Biology, not just over the whole world, but back through the centuries, or back through at least a century of time. So Theodor Boveri is a German embryologist, basically worked for 25 years between 1890 and 1900. At the time, embryos had been identified, had been studied. One knew you could follow the behavior and divisions of cells. One of the major questions was, where does pattern come from? Is it already present in some form in the egg cytoplasm, or is it generated at some point during development? Since only the mother contributes to the egg cytoplasm, any contributions from what was the sperm doing? How are these contributions? What is the role of the nucleus? If you look at Boveri's work, at the time, there was a general sense and an observation that the egg, when it was laid, had domains of cytoplasm that were visible. And a number of scientists had, at Boveri included, had traced the behaviors of different regions of the egg cytoplasm into different blastomeres and showed specific fates. One of the remarkable things about Boveri, and I've read through the literature, is the only of the papers that deal with this phenomenon, he's the only one that I can find who actually reversed the arrow and tried to follow the egg back into oogenesis to establish that the polarity and structures that one sees here, one can relate to the specific cells and specific positions in the ovary. That was an extraordinary insight into where this patterning comes from. But I think Boveri's most famous experiment on the organization of the egg cytoplasm was one that he did, an extraordinary, clever experiment that involved trying to establish what is the role of these cytoplasmic organizations. To do this, what he used were two different sea urchin species whose larval forms, or Plutia, had very clear and striking different morphologies. And what Boveri and what scientists wanted to know is what was the relative roles of the nucleus, and it was known that females and sperm and egg contributed nuclei to a large body of cytoplasm. What is the role of the cytoplasm? And what Boveri, to test this idea, used enucleated eggs from one species, so that only the cytoplasm was contributed for one species plus the organization of the egg, and then fertilized it with the sperm of the other species, and then asked is the final morphology of the embryo correspond to the information in the nucleus provided by the sperm, or is it provided by the egg cytoplasm? And the answer was that it looked identical to the sperm nucleus. And his conclusion was, and I'm quoting from the 1899, okay, this is his thesis work, 1899, at the age of 27. Herewith is demonstrated the law that the nucleus alone is the bearer of hereditary qualities. With the removal of material, the nucleus, with the removal of the maternal nucleus, are removed at the same time the maternal hereditary tendencies of the egg, and the maternal protoplasm, although in this case furnishing the large share of the material for the development of the new organism, is without influence on the final form of the embryo. I ascribe to the cytoplasm of the egg only the simplest properties responsible for differentiation. It provides the general basic form, the framework in which all specific added details are filled in by the nucleus. So, what Boveri is advocating here is that nuclear activity, you have to remember, DNA has not yet been associated, that it is the nucleus that provides a nuclear activity in the embryo that provides the specificity. But what was that nucleus? What was that information? You could see DNA in the nucleus, in chromosomes. One knew, and Boveri had actually proposed, that the process of meiosis and fertilization that had been described, the major job was to keep the amount of DNA constant. But what was that DNA, and what was their information content in it? And in a remarkable experiment of an insight into a tricky, difficult problem, Boveri chose to realize that it had been observed that polyspermy results in abnormal embryos. And the embryos are abnormal because the sperm, which would normally introduce 2 centrosomes, introduces 4, if you have 2 sperm, and that gives rise to a quadrupolar spindle. And what Boveri realized was that when this embryo divided, you had 3 sets of genomes being captured by 4 mitotic spindles and dividing. And what that meant was that you get different random combinations here, but statistically, individual blastomeres would have to lack specific chromosomes. And he did a whole set of experiments to show that it was actually the combination of chromosomes that was important, rather than the amount of DNA. And the conclusion was the only possibility remains, namely that not a definite number, but a definite combination of chromosomes is essential for normal development. And this means nothing else than that individual chromosomes must possess different qualities. In 2002. Now, 1902. It's still worth knowing about in 2002. Okay, so, but it went beyond this. So you look at these embryos, in principle, individual blastomeres are going to be lacking randomly different chromosomes. And what we realize when you follow these embryos, there's something interesting, that quadrants in the embryo became abnormal, and different embryos, because of the random distributions, became abnormal in different ways. And so that meant not only that chromosomes were different, but that individual chromosomes contributed specific qualities or specific information. All of the embryos they looked at developed normally to the blastosome stage, and then quadrants revealed themselves depending on which particular gene activity was deleted. So this meant and you could then begin to assign genetic information to specific processes. The idea that chromosomes were different from each other led Boveri, in a footnote in this paper, to propose an interesting idea. 1902 was right at the time when Mendel's papers had been rediscovered. And although one had Mendelian factors, as heredity, no one really knew where they were or what they did. But because based on the behavior of these chromosomes and their unique identity, Boveri suggests that he will, in a subsequent paper, and he did, propose that chromosomes are the carriers of Mendelian factors and Mendelian genetics. So all the way from the 1900s to around 1930 or 40, the idea that genes were on chromosomes was called the Sutton-Boveri hypothesis. One of the last and most interesting statements in this paper is the idea of development. This is 1902. Mendel has just been rediscovered, but people knew that nuclei were there and they wanted to be molecular biologists. The most important aspects, however, of the nucleus are completely inaccessible to an analysis with the present methods of physiological chemistry. In this respect, biology has at its disposal means of analysis of far superior resolving power. Even if the biologist is not capable of removing individual chromosomes, as would be the ideal case, he possesses nevertheless, in multipolar mitoses, a tool for the production of the most diverse combinations and provides an analysis of those qualities made possible by those combinations. This is a proposal to do developmental genetics, and I point out the challenge that you cannot be manipulated or controlled. So obviously from 1902, over the next 30, 40 years, genetics as a field was established through the work of Thomas and Morgan. And my second hero builds on that genetics and the availability of chromosomal rearrangements to address Boveri's challenge. Poulsen, among the fly field, is largely known because, as a graduate student at Caltech, did the embryology characterize all of embryonic development, the morphology of embryonic development in Drosophila. And for 30, 40 years, we would go to those books, his descriptions, to understand how development occurred. But Poulsen also did an important second experiment to address the role of chromosomes in development. The basic idea was to use chromosomal deletions and genetics to eliminate specific chromosomes in the embryo and then study the consequent development. And a particularly interesting case, the Drosophila embryo is missing the distal portion of the X chromosome. Ectoderm all develops into neural tissue. The most striking feature of such eggs is that they contain very little or no endoderm or mesoderm. The process of germ layer formation is interfered with seriously. The ectoderm proliferates, especially on the ventral midline, and produces what appears to be the semblance of an early nervous system. So deleting this region of the chromosome produced multiple defects, the most dramatic of which was conversion of all ectoderm, rather than becoming epiderm and neural tissue, into the neural pathway. But by using all the available deletion, Poulsen was able to break this phenotype and set up the standard for how we work, break this phenotype into components. The mesoderm degeneration could map to a different set of genes, but the neural phenotype could be mapped to a single gene, single band. This was an exciting result, having fundamental developmental biology associated with a single gene. And I'll point out to you that that gene we now know as Notch. And it was Poulsen who characterized initially the importance of the Notch signaling pathway. At the time that he did those experiments, it was unclear the relationship between DNA and heredity and the visible properties of development. Happily, the day is past when the view that genes have something to do with cellular physiology and development has to be fought for. From an embryological and physiological observation presented, it's clear that chromosomes are as much concerned with the process of development and in cellular physiology as they are with hereditary transmission. It's an interesting intellectual transition in our history. We went from heredity to the fundamental property of DNA. Another interesting thing in Poulsen's paper, the idea that genes in the nucleus would control properties of the cell was become gradually accepted But how they did that was unclear, and I want to point out to you that embryology played a central role in our understanding, specifically maternal effect mutations, because as Poulsen points out, to explain maternal effect mutations, where the effects of mutations are transmitted to the embryo even after the female nucleus is no longer present. The simplest hypothesis, and this is a little hard sentence to read, is that a non-diffusible plasma genes, or primers or something, controlled or synthesized by the chromo gene or nuclear genes, and are transmitted through the cytoplasm of the egg to the embryo. So the idea that you could have a nucleus and yet have an effect that was separate after the nucleus was gone led to the idea that the nucleus is producing something, RNA, which functions in the cell. Now, brilliant observations. I want to stop at this point and give you a personal memory of Don Poulsen. As a graduate student, I was an undergraduate, I washed fly bottles and looked at embryos and frogs and chicks, and I went to graduate school with the goal and the intention of becoming an embryologist and working on embryos that I knew about. But at the time, one didn't have rotations, and I ended up in Yale at New Haven and was assigned to Poulsen's lab because of my connections to washing Drosophila fly bottles back as an undergraduate. And I didn't have the heart to tell the man that I wanted to work on something with embryos, but it didn't take me very long to learn from him, the father of Drosophila embryology, that flies actually have embryos. I don't know where I thought they came from, Spallanzani and rotten flesh, but the essential idea that having learned, having the chance event transformed my life in that I got to look at fly embryos and saw that they were as beautiful as the frog and chick and sea urchin embryos that I had looked at before. Poulsen was in his 60s at this time and desperately did not want a graduate student. And my other memory of this was him retreating into his office often with me holding some reprint from the 1937 and trying to ask questions about the origin of the midgut. It took him about a year and a half, but he actually found a solution at the moment when a young assistant professor from Switzerland had been hired to the medical school, Yale Medical School, and for a variety of reasons, Poulsen was able to convince me that I really wanted to go over and learn some things from that lab. The lab was run by Walter Gehring. So I left Poulsen's lab and joined Walter Gehring's lab. So in a certain sense, I'm Donald Poulsen's last graduate student and Walter Gehring's first. His lab, Walter's, was empty when I arrived. Walter's vision of science at the time, and this was in the early 70s, was that molecular biology was coming. And in New Haven and then subsequently in Basel, Walter's goal was to transform this embryology lab into a molecular biology lab. And ultimately that paid off in terms of the spectacular results on gene localization that he'd observed. This is Walter, this is me. Walter's lab. For me, though, although I thought molecular biology was interesting, what was most important was, as is often the case, the lab and the lab mates that he had assembled there, and the people who I came to know, and probably the most singular, important person in that period was the short period of time that I overlapped with Christian and Usain Bohar. Obviously, our collaboration in Heidelberg gave us a chance to repeat Poulsen on zygotic gene or repeat Boveri. And you can see our screens were designed to identify zygotically active mutations that transform the embryo. This involved establishing large numbers of mutant lines and looking at lethal mutations where homozygotes, interesting mutations and working these down to genes. In this screen, although these all involve genes which must be transcriptionally active, we also had lines where there were no lethal mutations. And so we actually, an initial probe screened through the non-lethal lines to look for maternal effects, and we found a small number telling us that this was possible, and it was possible to identify maternal effects. Now, subsequently, Jan Uslang and Trudy Schippbach in Princeton carried out massive scale screens of the order to identify and define these maternal genes. What I want to use now is to take these two screens, put them into a context, and say, what is the general picture that emerged? One of the, one of the conclusions is that most RNAs and proteins that you find in an egg are supplied by the mother. But because they're needed at other stages, they cannot be easily studied genetically. The only thing that can be easily studied is things which are specific. The embryo uses zygotic transcription. This is my intellectual summary from the screens to control spatial distribution of a small number of gene products whose function switches, whose levels control cell fate. So the presence or absence of gene products and the fact that you can use transcription to do that is central to the roles of these genes, and transcription does that for you. Small number of maternal effect genes establish gradients that control the pattern of transcription. I'll remind you of Boveri's comment, and we can ask, how closely does this particular organism match? And Boveri's view, it's interesting, it says that cytoplasm, small, provides basic framework, basic form, framework, which all specific details are supplied by the nucleus. So in a certain superficial sense, it looks the same. My own sense is that Boveri is wrong, or not wrong, or we don't know the relative importance of maternal and zygotic effects. In my own lab at the moment, one of the things that seems to arise from more detailed analysis of Drosophila is that maternal cytoplasm provides much more than a basic framework. In Drosophila, we've been able to show, collaboration with Thomas Greger, when he's a graduate student, that the bit weight gradient is extraordinarily precise and reproducible and defines positions, single cells different from their neighbor, reproducibly. The information is there, and more recently we've been able to show that zygotic genes appear to take this precise maternal, the cytoplasmic information, and their role of transfer is not to invent or generate information, but instead to translate information that was present in the cytoplasm into a language of gene activities that control fates. That's a model. It emphasizes the importance of maternal effect mutations and puts zygotic transcription in a passive response. To address that, I'm just going to end in the last 2-3 slides with one of my own personal experiments, and was to try to begin to test that. And the idea here is to say, well, what would happen if we eliminated all maternal information from the embryo and only left the zygotic component? What do the embryos do? How can they, what do they do? To do this is all obviously possible because by eliminating maternal effect information, you flatten the gradient, but there are enough alleles that allow us to adjust the levels such that we can dorsalize or change the dorsal ventral fates of cells uniformly along different levels along the dorsal ventral axis that are the anterior posterior axis, and we can create genotypes which are uniform but have different levels and would correspond to different positions on the embryo. So the idea is to make uniform embryos, all of which are composed of the same cell type, and then ask, how do these embryos develop? The kinds of questions that I'm trying to are they really uniform? And the answer is yes. What does that uniformity mean? In some cases, they're uniform in meaningful ways in that the transcription factors that are expressed are representative of specific regions in the embryo. In other cases, we can generate initial states that are slightly off of anything that exists in the embryo, and then ask, how does the embryo, do the circuits drive you back to meaningful states? Are the states meaningful? Are they maintained? How is proliferation controlled in the absence of pattern? Do these embryos undergo proliferation? Are there clocks which drive events that are independent of specific programming? And one of the things, interestingly, we've observed is that these embryos, although they initiate uniformly, don't necessarily always remain uniform. In that patterns arise, and these patterns, for the moment, appear to be random with respect to axes of the embryo. We're trying to figure out what the rules are associated to characterize these patterns. Now, any of you who know me would know that I wouldn't just stop looking at a bunch of dead embryos. My particular love of obviously has been watching gastrulation. And so one of the things that's kind of interesting for me at the moment is to follow and compare. If you have no dorsal ventral pattern, all the cells are uniform along the dorsal ventral axis. They still have anterior posterior pattern, and you get certain kinds of defects that are extremely reproducible and that I want to understand. If you have no patterning at all, you might expect that you can't have movement, because you must have differential force generation to produce movement, and yet things happen in these embryos that I believe will be informative and present us with opportunities for understanding the process. Now, okay, I'm going to stop. We are living in a remarkable time in our field. We can't but be enthusiastic about the technologies that we have available to us now, the things that we can see, and the opportunities that we have, even in weird quadruple mutant, and to use weird quadruple mutant embryos to begin to try to understand. We are at a time where we can address fundamental questions. So, getting back to this issue, the social issue, since we've been doing personal comments and personal stories, I'll tell you my personal dream, the impossible dream that carries me through very often, that something that I cannot have, but I would love to have, is to go back and talk to Theodor Boveri and talk to Donald Poulsen and tell him, tell Boveri, tell Poulsen what we know now. The things that they worried about, the things that they thought about, the wonderful discoveries that they made. Our understanding of them have changed in ways that would matter to them. And this desire to this imaginary moment that I've lived through so often in my experiments, where I say, I want to go to the door of Don Poulsen's office and tell him this thing. We don't have that opportunity. And so I think that one of the things at a meeting like this, why it's valuable, is that we don't tell our mentors, we don't tell the past, but we can tell the present and we can influence the future. That, I think, is our goal. And it's been a wonderful pleasure for me. Thank you.